Hey y'all, my name is Prashanth Kumar, or PK, and I'm an MCAT tutor at Shemmassian Academic Consulting. Today, we're doing a passage walkthrough in the MCAT Biochem section. You should be taking roughly 8 minutes on this passage, but today we're going to be going more in depth so you can see exactly what's going on in my head as I tackle this section. Let's get it. Alright, great. So I've gone ahead and put the passage on the left and the questions we're going through on the right. So feel free to take a moment, pause the video, and come back to us. I'll go ahead and zoom in on the passage just so you guys can get a better look. Great, and now onto the questions. Awesome. So with that, let's get right into talking strategy. I like to maintain the same exact strategy going into any passage. The reason for this is I have a routine I know I'll go into test day with, regardless of whether the passage is super easy or super hard. I know that I can come back to my roots, and that builds confidence. Confidence saves time, and saving time, higher score. So for me, that strategy is read a paragraph, write a quick 3 to 5 word summary, then go on to the next paragraph, do the same, and on and on and on. The reason I like doing this is that it provides accountability at the end of the paragraph, making sure I understand exactly what's going on before I move on to the next paragraph. I often found myself reading an entire passage, sort of skimming in my head, not really understanding. And at the end of the passage, I'd have to reread the entire passage, and that's a huge waste of time. Here, if I don't understand the paragraph, I can just reread it really quickly and then write my 3 to 5 word summary. The summary just is a quick check-in, making sure I understand exactly what's going on before I move on to the next one. Additionally, I don't like spending too much time on figures on that first read. I think that the figures will be asked about in a specific way, and we'll need to come back to them if a question asks about it. But sometimes the figures aren't even asked about, so it doesn't really make sense to spend a lot of time going through all the details of a figure if it's not going to be asked about, right? We got to save all the time we can. I also don't highlight too much. It's not really for me, but if it's for you, feel free to. But with that, let's get right into the first paragraph. The endoplasmic reticulum, ER, is a multifunctional organelle and plays a crucial role in protein folding and lipid biosynthesis. Proteins synthesized in the ER are post-translationally modified via N and O glycosylation. The SEC59 gene encodes a dolichol kinase that is required for N-glycosylation in the ER. Researchers are interested in studying the role of SEC59 created and characterized at SEC59-1 deficient cell line. All right, so the question I always ask myself is, what's going on in this paragraph, right? The central idea, a thesis in a sense, not really focused too much on details, just a quick summary. And so here, we're talking about the ER and its role in glycosylation, and specifically looking at genes. And so right away, all I'm going to do is, okay, let's write a quick summary. So we're looking at SEC59 and its effect on glycosylation. Good. And so with that, we have our quick summary. And we're ready to go on to the next paragraph, right? I don't care about every single detail. We'll come back to it if needed. In experiment 1, researchers created a deletion in SEC59-1 and analyzed levels of phosphatidylcholine, phosphatidylethanolamine, phosphatidylinositol, and phosphatidylserine. Equal amounts of WT and SEC59-1 delta cells were grown in YPD medium at 30 degrees Celsius. Equal amounts of cells were harvested in a time-dependent manner. Lipids were extracted, phospholipids were separated by two-dimensional TLC, and the amount of phosphorus was quantified. The results are shown in figure 3, and then it gives us a figure, right? So right now we have an experiment. So I'm just going to put a 1 right there, indicating experiment. And what is it looking at? Well, it's looking at wild type versus SEC59 delta cells, right? And these different phosphatidyl groups, right? So I'm just going to do P blank on the effect, right? A little arrow. And remember, this might not make sense to anyone else. All that matters is it makes sense to you, right? Use all the abbreviations, symbols, arrows, whatever makes it easier for you to understand. Use whatever you need to. No one else is going to be reading this at the end of the day except you. So I clearly use my abbreviations, and it might not make sense to the next reader, but it makes sense to me. All right, and so remember I said we have a figure. We're not going to spend too much time looking at the figure. All we can look at is the y-axis is looking at phosphorus, and then the x-axis is looking at time, as well as these groups. So just a quick glance, nothing really crazy. and we'll come back to it if we need to. Researchers then hypothesized that an overexpression of SEC59 might also have an effect on lipid composition within the ER. To overexpress SEC59, researchers cloned the gene into a YEP352 vector. In experiment 2, the researchers also investigated phosphatidic acid, PA, lysophosphatidyl-choline, LPC, and cardiolipin levels. Researchers investigated wild-type cells, cells with an overexpression of SEC59 through the addition of YEP352 vector, SEC59-1 delta cells, and SEC59-1 delta cells rescued by YEP352 vector exogenous SEC59-1. The results of this study are shown in figure 4, and then we have another figure. All right, so a lot of details, a lot of words, knockouts, so many cells. All right, we're not going to focus too much on it right now, right? We're just focusing on big picture understanding. Okay, so now we're looking at the overexpression of SEC59. So this is sort of experiment number 2, so I'll put 2 in a little box. And so we're looking at increase SEC59 resulting in question mark on P blank additionally, right? Plus more, plus etc., right? Something else. We're looking at increased SEC59 levels on these phosphorus levels in these other groups. And so how are we going to do that? I'm going to draw a direct arrow. I'll put a question mark there, but a direct arrow as a result of YEP352, right? So we have a big picture understanding of YEP352 is increasing the amount of SEC59, and we're going to look at that effect on these phosphate groups and cardiolipin and whatever other groups, okay? And so with that, we see the same exact structure in this figure. We're not going to spend too much time on it, right? Phosphorus on the y-axis, all these different groups on the x-axis. We see the same like table of different types of cells, but not really focused on that. And we're ready to go on to our final paragraph. In experiment 3, researchers determined that dolichol kinase-dependent protein glycosylation directly or indirectly plays a role in suppressing beta oxidation. All right, so what is happening? This is a very quick paragraph, so it's just going to ask for a little summary. So I'm just going to write dolichol kinase glycosylation. I'll just abbreviate. Dolichol kinase glycosylation results in suppressing beta oxidation. So I'm actually going to change my arrow from a regular arrow to a flat arrow. Let me erase that real quick. So we have a flat arrow to indicate suppression. So these dolichol kinase glycosylation suppresses beta oxidation. And with that, we are ready to go straight on to our questions, right? So now we have a big picture understanding of everything that's going on. And if we need to, we can come back to it. Remember, the summaries are used as a table of contents in a sense. You don't have to come back to it if you need to, if you don't need to. But if you do, you know exactly where you're going to be looking for these details or this information. So right away, let's go number 1. Which of the following is the most reasonable conclusion based on figure 3? Wild-type cells lack adequate levels of ER phospholipids. Cells lacking SEC59-1 contain higher levels of phospholipids compared to wild-type. SEC59-1 acts as a transcription factor to repress phospholipid expression. D, SEC59 is the primary repressor of phospholipid overproduction. Okay, so right away we know exactly where we're looking, right? We're looking at figure number 3. And so this is why I said earlier, we don't need to go back to the figures unless the question asks for it. And here's an example of that. And so we're looking for some sort of conclusion of that. So let's go back to figure number 3. And so figure number 3 is asking us to take a look at the difference between the wild type group and the SEC59-1 delta group, right? Because all of our questions, our answer choices are asking us about SEC59. So the important thing is anything with a delta after it, the delta means deletion, right? So we should understand that is very, very important, okay? So in this case, SEC59-1 is deleted, and so we're looking at the effects. And so generally, when we're looking at this, I want you to look at trends, right? Graphs are often complicated. Experimental design is messy. And so I want you to look at general trends, namely between one group and another group on the MCAT. And so here, our 2 groups are listed because they're in the legend, right? We have our wild type group or our normal group and our sort of deletion group, which is our other group. And we're looking at trends between them. So let's take a look at the first one, right? So let's say we start with the wild type group for the PC. So that's right here. I'm going to use a little laser. So that's right there, right? And so what happens when we delete that for the PC group? We see a massive increase. In fact, we see these 2 stars indicating that it is statistically significant, right? And that's very, very important. The stars mean that something is different is happening. And so we're looking throughout the chart or figure, and we see all these arrows are continuous, right? Regardless of whatever group we're looking at, they're all going to have these significant, right? Significant changes. And so this is illustrative of a trend, right? When I'm saying look at one group versus another group, I'm not looking at every single data point, how much they're increasing. I'm just saying, there's a really large increase and it's statistically significant. So that's exactly what we're going to use as we go here, right? So let's go through answer choice by answer choice. Answer choice A, wild type cells lack adequate levels of ER phospholipids. That doesn't really make sense, right? Wild type cells lacking adequate levels? Cross that out. Wild type cells should have normal levels of ER phospholipids. So we can already eliminate A. B, cells lacking in SEC59-1 contain higher levels of phospholipids compared to wild type. All right, so our question here is asking what happens when we decrease SEC59-1? Do we have increased amounts of phospholipids? Yes, exactly, right? When we delete the SEC59, when we delete it, that results in decreasing SEC59, right? And so when we delete it, we have decrease, but we see an increase in the amount of phosphorus. So decreased SEC59 results in increased phosphorus. And so that means we have increased phospholipids. And so right away, B is looking like our best answer. SEC59-1 acts as a transcription factor to repress phospholipid expression. Okay, so that would result in increased SEC59 acting as a transcription factor. We don't know anywhere where it says that SEC59-1 acts as a transcription factor. So that could be true. It also could not be true. So we can eliminate C right now. D, SEC59-1 is the primary repressor of phospholipid overproduction. Again, nowhere do we see it's the primary repressor, right? It could be, it could act as a repressor via transcription factor, via not a transcription factor. But remember, we're working in the confines of our data as well as our passage. And nowhere does it say that it's the primary. Primary is like the main thing that represses it. We could see another sort of gene or another sort of protein or another sort of transcription factor that's deletion is the primary repressor. So we don't want to infer beyond the passage. So right there, we eliminate D. And B is a simple answer, and never be afraid of the simple answer. We're going to stick with B because it tells us exactly what the trend is. And with that, we're 1 for 1. On to question number 2. According to results from figure 4, what effect does SEC59-1 overexpression have on phospholipid levels? Is it A, increase in phospholipid levels, B, decrease in phospholipid levels, C, phospholipid levels remain constant, or D, phospholipids are shunted into gluconeogenesis? All right, so right away, we know exactly where we're looking because the passage tells us. And so we're looking at figure number 4, and we're looking at specifically SEC59-1 overexpression on phospholipid levels, right? And so how do we result in overexpression? This is something I want you to remember because we wrote it right on our table and contents, right? We said that this YEP352 results in overexpression of SEC59, right? And so we're looking at our gene with the YEP350, our wild type with the YEP352. So we have our wild type here. That's our normal or sort of nothing modified. And we're going to compare it to the YEP352 plus wild type group. So we're looking at this dark red and this dark checkered green. And so right away we see that's the first 2 of the 4 for each of these, right? And so remember what I said. We're looking for trends. We're not looking at value differences. We're looking at general trends throughout the entire thing. And right away when we're looking, we don't see the stars, right? We saw the stars in the previous question, but there's no stars in between our 2 groups that we're comparing. So we're not seeing anything statistically significant. That's very important. Additionally, I want you to always remember that these are sort of the bars that they see up here tells us sort of the standard deviations or the variance of our bars. And so if the bars are overlapped, generally that's a good sign saying these aren't statistically significantly different. If they are completely separated, you can usually say, okay, this is a good sign. Always look for the stars, but this is a good sign. So possibly significant, all right here. and I'll write not significant right here. Okay? And so with that, we should know that we're not going to see an increase or decrease since they're right around the same level for each of those groups, but they remain constant. And we don't see anything about them shunted into gluconeogenesis because we have the same amount. And so we're left with C as our best answer choice. We're inferring the amount of phospholipid levels based on our data. And so with that, we're 2 for 2. On to question number 3. Let's take it again. In a new experiment, researchers used 32-phosphate. What is the resulting element if the phosphate undergoes alpha decay? Is it A, 28P, B, 30P, C, 28Al, or D, 30Al? All right, so right away, we're looking at alpha decay, and we should be having alarm bells ring in our head. We're looking at sort of nuclear decay or nuclear reactions, specifically around 32-phosphate, right? And so what does that mean? What is alpha decay, right? Decay is when we have some sort of molecule being released by our substance of choice. And so alpha decay is when our molecule emits an alpha particle, which is just a helium. It's a 4-2 helium, right? So this alpha particle, is being decayed. So we're turning 32-phosphate into helium 4-2 plus something else, right? Plus, I'll just write X right for now, right? So we're turning this particle into 2 different particles, our alpha particle helium and our X, which we don't know yet. Okay, so right away we need to know how to do these problems is basic math, right? It starts with just basic math. And how do we do that? Well, I like to say left equals right for top and bottom, right? So what that means is I'm just going to write A and B here, okay? And we know phosphate, we got to look at our periodic table, and the atomic number is 15. So let's start with that, right? How are we going to do this? We're going to say left equals right on top and bottom. So let's start with top. So 32 equals 4 plus A. I'm saying the top, right, 32, 4, and A, on the left and right have to be equal to each other. So what does that mean? That means A is equal to 28. Okay, so right away we can eliminate B and D because we know the top has to be 28. And we're going to do the same exact thing on the bottom, right? We're going to say, okay, 15 equals 2 plus B, right? Left equals right on 15, 2, and B, okay? And so what is B equal to then? B is equal to 13. So now we know that our X is going to be atomic number 13 with a mass number of 28, okay? And so right away, we should know that if the atomic number changes, the molecule changes itself, right? So phosphate is no longer going to be phosphate. So it'll turn into aluminum. And so we have our aluminum with a mass number 28 by this nuclear reaction. And so with that, we are 3 for 3, and we have a little bit more understanding of nuclear decay with alpha particles, or alpha decay. All right, question number 4. Researchers observe an increase in acetyl-CoA in SEC59-1 delta cells. Based on information from the passage, which of the following best describes this observation? A. SEC59-1 deficient cells produce higher levels of phospholipids, which are converted into acetyl-CoA through beta oxidation. B. SEC59-1 deficient cells must decrease flux through glycolysis in order to conserve energy. C. Acetyl-CoA accumulates as the Krebs cycle stops. D. Cells with SEC59-1 can only use phospholipids for energy production, causing beta oxidation to occur more rapidly. All right, so right away, lots of different things being brought up, right? We're talking about our SEC59-1 delta cells, but it's also talking about acetyl-CoA for the first time in a while. And how do we know acetyl-CoA is linked to this? Well, we're going to look at our final paragraph, right? It talks about this glycosylation as it results into beta oxidation. And we should know beta oxidation is the idea of creating more acetyl-CoAs when we're in the state of energy dependence on fats and breaking down fats, right? And so what's happening, right? We're saying, okay, if we're going to increase the amount of acetyl-CoA, then we're going to have some relationship to its effect on beta oxidation. And so right away, we can eliminate B and C because we're not talking about glycolysis or Krebs cycle at all. We're focused only on beta oxidation. And so we can look at our sort of diagram or table of contents, and we see that these kinases inhibit beta oxidation, right? These kinases inhibit beta oxidation. And these kinases are found in these SEC59-1 cells. So what happens when we, I'll draw that like that. What happens when we knock out SEC59-1? We knock out the kinases and we knock out the suppression, right? So if we knock out the suppression, we're going to see an increase in beta oxidation, right? So let's link it together again. We're saying kinases are inherently, these kinases that we discussed in the lab, the dolichol kinases, are inhibiting or suppressing beta oxidation. So when we have SEC59-1 delta or knockout, we're going to remove that kinase, we're going to remove that suppression, and we're going to see an increase of beta oxidation, right? And so right away, we're down to having more beta oxidation, which A and D do say, okay? Oops. Sorry, I'll undo that. And so now we can use our trends from our graphs to help produce the last part, right? So it's saying SEC59-1 cells produce higher levels of phospholipids, right? Is that true? Well, let's remember, right? When we knocked out SEC59-1 alpha, what did we see? We saw this increase, right, in every single type of phospholipids. And so that is going to indicate that A is the best answer. A is saying we are taking these deficient cells, we're removing SEC59-1, and we're, as a result, increasing the number of phospholipids. How? We're saying it's converted through a beta oxidation, increasing in this case because we remove the suppression. D, cells with SEC59-1 can only use phospholipids for energy production. So we always have to be careful with this saying only. Remember, MCAT hates absolutes, but they always try to trick you with that. Anytime it says only, you can pretty much eliminate it unless it's an extreme situation, right? We never see SEC59-1 saying we only use phospholipids now for energy production. Nowhere is that suggested, nowhere is that claimed. And so as a result, causing beta oxidation to occur more rapidly, while that might be true, while we might see increased beta oxidation, it's not for that reason. So we can eliminate D, and we are 4 for 4. All right, on to our final question of the day. All right. Question number 5. The YEP352 plasmid encodes a gene for ampicillin resistance. Researchers treating a YEP352 transformed bacterial colony with ampicillin should expect A, surviving cells to have lower levels of PC when compared to PC levels in untransformed cells, B, surviving cells to have similar levels of PE and PI when compared to PE and PI levels in untransformed cells, C, some YEP352 cells remain sensitive to ampicillin, D, acquired resistance to develop rapidly in cells with the YEP352 plasmid. All right, so right away we're seeing, okay, we have this YEP352 that we talked about earlier. It's encoding a gene for ampicillin resistance. Okay, so what does that mean? That means it'll be resistant to ampicillin, as it makes sense, right? And so if we see ampicillin cells, they will no longer be sort of sensitive to ampicillin. They'll be resistant. Okay, so with that, we can remember where we talked about YEP for the first time. Well, we talked about YEP in this final paragraph, right? In that, we were adding YEP and seeing the effect on phospholipids, right? And what did we say? From question 2, we should remember that when we added YEP, we saw no changes, right? It was no change throughout because there was no statistical significance stars. So with that, we should note that no effect on phospholipids. That should be the first thing we look at, right? And so we can eliminate A almost immediately, and we clued in on B being similar, right? We have similar levels of PE and PI and PE and PI. So right now, B is looking like a great answer. So let's go to C. Some YEP352 cells remain sensitive to ampicillin. This cannot be true. if we have the gene coding for ampicillin resistance encoded with the YEP352 cell, any cell that has YEP352 will be resistant to ampicillin, not sensitive. So we can eliminate C because it's saying it's sensitive. And D, acquired resistance developed rapidly in cells with the YEP352 plasmid. This is not a case of a acquired resistance where we're gaining resistance to something. Once we have the plasmid encoding, it's resistant. So acquired resistance is not the correct term here, and so we can eliminate D. And so we're left with B, and so surviving cells will have exactly similar levels of PE and PI compared, and we're 5 for 5. We crushed it. All right, great work, team. We crushed another biochem passage. Kudos for you for getting it done. Lots of great stuff from experimental design to radioactive decay. So great job. If you like this video, give us a like and comment down below exactly what you're looking forward to next in terms of content or walkthroughs, and subscribe to our channel. Additionally, check out our 528 strategy series from Vikram. Super helpful strategies for each of the sections to help you get your dream score on test day. And finally, click the link in our bio to sign up for the MCAT question of the day so you don't miss out on any test prep before test day. Happy studying, and let's get it.